you know, dry, wet, tipsy, and then hot spring. Yeah, just so many resources that we can, explore uh, our microorganism potential from. And as we know that also in BRIN, to support this kind of research, we have provided several scheme, Prof. Soon, that for example BRIN also run a collaboration platform, expedition, and also we have several activities, join cruise with Chinese government to explore microorganism. One of its microorganism from the sea, from the deep sea. And then, and also we have scheme of, we call it research collaboration center. And currently our research center, we have collaboration with Undip, Diponegoro University Central Java, get grant with the topic of thermophilic enzyme. I think it is very interesting how we can not just, knowing that we have so many diversity, but also we want to know more and understand more how we can explore, how we can find them, how we can bring them to the surface, to the laboratory, and to utilize them. Yeah. So this is very interesting yet challenging, Prof. Because we can just count by our finger how many researcher in BRIN that focus on this kind of extreme environment. Not many. Dr. Gilda, and then we have several, less than 10, less than 5 probably. Yeah. So, and actually this year we are also proposing uh, to the management to develop extremophile laboratory. Yeah, because it is very important, and we still don't have like a proper room or laboratory to conduct this kind of research. Yeah, hopefully by your supervision and by our collaboration. Yeah, so everyone, actually we have our center. Yeah, we have signed the agreement, collaboration agreement with Prof. Sun institution, regarding the discovery and development of bioactive and also enzyme from the microorganism. And hopefully he also can give us many inputs on how we can not just conduct but accelerate this kind of research in the area of microorganism from extreme environment. So again, thank you, Prof. Chun, that you will, as a dean, you are coming to BRIN, and then giving us many knowledge and also technology that you may also later on transfer to our researchers. And again, we hope that we can have like strong and long collaboration between BRIN and your institution regarding microorganism research. Yeah. So everyone, so please enjoy the webinar that hopefully we can, have a fruitful discussion with Prof. Soon. And also, if everyone here is interested in this kind of research, you may also contact directly to our PI, Dr. Gilda. She's one of researcher that focus on extreme environment research. But actually, Prof. Sun is here with Dr.rer actually as a host for a visiting researcher. Yeah. Okay. I think that's all from me, Dr. Zida, and everyone. Thank you very much. And by Bismillahirrahmanirrahim, I also officially open this webinar. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Bu Julia. I give the screen back to you. Yeah.
Thank you, Fathoni, for the opening remarks. I forget to mention to all of you that Professor Chenghang is a visiting researcher hosted by Dr. Handayani. And actually, Professor Sun won this seminar to be offline, because offline you can discuss more detail about the research and also the future collaboration. But it's okay if after the presentation and then you interested to ask more question or collaboration, Professor Chenghang soon will be in Taxa laboratory till Friday afternoon because he leave Jakarta on Sunday afternoon. So please welcome to Taxa laboratory
Sunday,
and
Bu
have
Dewi?
discussion.
This Sunday?
What?
This Sunday? Next Sunday?
Yeah, Sunday. This Sunday.
Already 1 month,
This
is it?
Sunday. This Sunday in a very early morning on Sunday.
Oh, okay. So I will try to visit laboratory on Friday. Yeah, I'll try. Yeah,
I plan to
yeah,
take Professor soon to your office,
okay.
Fathoni, maybe on Thursday to say
Thursday
goodbye.
or Friday, yeah. Thank you. Yeah.
Okay. I will make appointment with your secretary. Okay, ladies and gentlemen, let's start the seminar and I will give the screen to the moderator, Dr. Ira Handayani. Mbak Ira, silakan.
Yeah, thank you, Bu Zilda. Good afternoon, everyone. Welcome to today's webinar titled Micro from Extreme Environments, What We Do and What We Can Do. The Thinking of the Future Research Project. My name is Ira Handayani, and I'm honored to serve as your moderator. We are delighted to have with us our chairman, Mr. Fathoni, and a colleague from the Research Center for Applied Microbiology, researcher from BRIN, and of course, our students who are joining today. Thank you very much for your presence and participation. It is my pleasure to introduce our distinguished speaker, Bu Amalia from Team Shopping. Could you please share the CV of Professor Chenghang? Okay. Please make it bigger. Oh, I see. Okay. Thank you. Let me introduce Professor Chenghang. Professor Chenghang, Dean of the Department of Microbial Chemistry, the Institute of Medicinal Biotechnology, Chinese Academy of Medical Science. Professor Sun earned his PhD in the Microbial and Biochemical Pharmacy in 1999 from the Institute of Medicinal Biotechnology, China's Academy of Medical Science and Peking Union Medical College. He then carried out postdoctoral research in 2000 with the Nobel laureate Professor Satoshi Omura at the Kitasato Institute, Japan, followed by another postdoctoral fellowship in 2002 at the Bloomberg School of Public Health, Johns Hopkins University, USA. His research lie in the bioprospecting new antibiotic-producing strain, particularly bioactive actinomycetes isolated from extreme and hard-to-reach environments such as karst, caves, hyper-arid deserts, salty lakes, the Qinghai Tibet Plateau, and also mangrove ecosystems. Professor Sun's pursuit of the new antibiotic has led him to build many international collaboration and contribute globally to search for novel microbial natural product. Under his leadership, his group has published over 100 peer-reviewed academic papers covering the chemistry of microbial natural products, systematic taxonomy of actinomycetes, microbial metabolomic, and also genomic. We are special honor because Professor Sun is currently joining us here at the Research Center for Applied Microbiology BRIN as a visiting researcher for 1 month. His presence here is a wonderful opportunity to strengthen collaboration and exchange the ideas. Without further delay, please join me in welcoming Professor Chenghang Sun. Professor Sun, the floor is yours now.
Okay. Can I share my PPT?
Okay, thank you.
Selamat siang. I'm Chenghang from Peking, China. Thank you very much for Tony's introduction, for Dewi and Ila host me here for presentation. Thanks you all for participating in my presentation. Today, I will give a brief talk about extreme microbials from extreme environments. Because before I come, we just sign agreements and prepare a joint research for joint laboratory supported by the Beijing city. So we are waiting for the results. Hopefully, we can get the support from government and hope we can continue and long cooperation each other for microbes, because our institute in China, which was focused on the actinobacteria, and Indonesia, BRIN, our institute here, actually applied microbiology research center, so which was very fit each other for future cooperation. And thank you, Tony, for approve my proposal on Side 4 and give us very strong support for possibly future research project. And Ira and Devi take me a many place to visit our laboratory and also different facility, which make me more confidence to do more research with the Indonesia scientists. Okay, let me begin my topic today. I will talk according to the contents, which I will briefly introduce our institute and also Chinese Academy of Medical Science and Peking Union Medical College. Also, I will give some example which we have done in the extreme environments. Finally, I will give the international cooperation grants in China and Indonesia. Actually, in the 2018, at that time, I tried to find the grants, but very few. But now, many and many grants appears between two governments. So the Ministry of Science and Technology have many grants cooperation with BRIN. So hope we can get one or two to continue our research. Let me first briefly introduce the Peking Union Medical College. Peking Union Medical College actually was founded in 1917. Actually, it was more than 100 years before, at that time, still Qing Dynasty. This institute actually was the first Western-style medicine founded by the Rockefeller Foundation. Till now, 100 years after, it has become the #1 university of medical research and education. So every year, there are more than 3,000 PhD and master students graduate here. We can divide in two parts. One part was like 120 scholar, like bachelor scholar students will join that university, which was very unique because they has an 8-year study, then directly get PhD degree. This for 120 students. Another part was from all over China, was master students and PhD students. Usually PhD students from master, it takes 3 to 5 years. And from bachelor to the PhD, usually 5 years. And if you want to get the master degree there, you must have 3 years master degree study. So this was a kind of unique in medical university. And also this university was merged in 1957 with the Chinese Academy of Medical Science. Now they become the same research center. In the 1956, Chinese Academy of Medical Science was established, and then merged with Peking Union Medical College, become the only medical research and education unit. So until now, they still keep this system. One was education, one was like research. Also, they have more than like 10 hospitals, which was each hospital is almost the top in China. So they have institute, hospital, and the university. Even the Peking Union Medical College has ever changed the name as Peking Union Medical University. But in the medical field, no matter in domestic and internationally, they think Peking Union Medical College is more famous, and everybody knows that. So we changed the name again, become the Peking Union Medical College. In PUMC, actually, there are 3 medical-related institutes. One was IMM, another was IMPLAD. IMM, which focus on the nature products and the chemical synthesis, which from the plant or animal. IMPLAD, which was focused on the identification of herbs, and they have like bar, like plant ID of each Chinese herbs. So they focused on different fields. In my laboratory, in my institute, actually, we focused on the nature products from microbials, especially from actinobacterial, because actinomycetes produce 90% clinic medicines from microbes. Actually, the first penicillin was how to say screening, become the commercial products in China. So in that time, actually everything was low primitive, and no matter from laboratory or factory, is very, very simple. Now, after 60 years, many things changed. Even though we have changed like 3 times names, now name is the Institute of Medicinal Biotechnology, but most of our research is still focused on the discovery and the development from antibiotic from actinomycetes. When I first visited Tony's lab office, I have a book as a gift. Actually, that was 60 years celebration for establishment of our institute. Now more than 200 professor and assistant professor, associate professor in this institute, almost like double students. So totally 600 people in our institute. They have, during this more than 60 years, many antibiotic, no matter it's Me Too or innovation kind of products was found and developed in our institute. For example, we have methyl vancomycin, which against the MRSA used in the clinic. Also, we found some johnstonemycin, which was used as an agriculture antibiotic against the plant pathogenic fungi. Of course, we have gentamicin, which was helped animal healthy and grows faster. But unfortunately, the yield is not so high, so we have to give up this compound. Microbe actually was one of many units in our institute. We are the source of new antibiotic. So we have strength collection center. They focus on all kinds of strengths. We've only focused on actinobacteria from extreme environments. So as we know, penicillin is the #1 or the very beginning of antibiotics, but it's discovered occasionally, luckily, by the Fleming, not like Waxman. So he set up Waxman's method for discovering new antibiotics. And all over the world, scientists use this method, produce many new antibiotics used now. But after the 1970s, it became quite difficult to get new one because used this way, many new antibiotics and many actinobacteria already being studied. Even though now we already set up many new methods, such as multiple omics, and also we try to find new sources of actinobacteria. But after several years study, we realize even though we use all those new technology, found old one is unavoidable. So how to get producing strain which has never been studied, which was a very notorious question and the problem and the bottleneck for new antibiotics. So how to, in the very beginning, the replication of producing strain and the replication of chemical compounds is a very important issue. We found if we get a very good producing strain, which has very rare species, actually they can produce many new compounds. For example, these strains we collect from desert, which produce more than 40 compounds. Most of them was new. We even double this type of compounds. For example, before there were only 12 aldegamycin already be discovered. Now we found another 16, which far more than non-antibiotic get together. So if we get a very good strain, then we will get big progress. So how to get the unique strains is what we always thinking. So there are several ways which can help us to get this one. The first one, we still use the traditional cultivation method, but to very hard-reached environment, we say unexplored environment. Another was we can use some new technology to try to get very unique strains. Recently, I'm not sure you guys have already paid attention to their research of Kim Lewis and Slava Epstein. Actually, these two scientists, they make a novel biotic pharmaceuticals, which many how to say invest money go to their company. Epstein actually he was a Russian marine scientist. He go to the United States from the postdoc and then finally established iChip technology. I'm not sure everybody know iChip technology because they make like more than 96 well, very small volume, and they can put agar and sample into this very small hole, and they put the chips go back to the environment to catch up the strains which never been found in the laboratory, because he used the great plate count anomaly, this kind of phenomena, and put all the iChips go to the in situ soil, which can get more growth factor, which cannot be get in the laboratory. So that's why there has many more strands can be growth rather than only we use ISP2 or RR2A, this kind of medium. After this technology, many new antibiotics was discovered in their lab. Except iChip, also other method was now used in obtain the unique strains. For example, in our laboratory, we designed this kind of equipment, which actually was two membrane and with agar. Then we glue it inside already some nitrogen was there. So we embed it in the mangrove. So the strands, after 1 month, we take them off and go back to the laboratory. We can find some strands which was never found before. That's most was new species. So this system was my PhD student's work. And we also found some active strands, which we are doing chemistry now. Exactly this new technology, and also many group, they go to the extreme environments to collect sample to find very unique producing strands. So like deep marine was one of the this kind of research. Now people already can go to Mariana Trench to get sample. And also, this one was done by the Canadian scientist. I put this as an example because I observed many papers, the volcano, which was quite a popular study right now. And till now, there are no many results yet. And in Indonesia, the volcano, actually many more volcanoes. So if we go to the Google to try to find a good result, it's still very few. So in Canadian scientists, they go to the Turkmenistan, they go to the Darvaza gas crater. I guess some people know this place because in the 1970s, Russian scientists go to there to find gas, and they put some fire. Until now, they didn't stop yet. So this place, no research on the microbes. So Canadian scientists, they make and find clothes which can resist the heat, and put a kind of lime in the like a crater, and they downstairs to collect sample here, different place. And actually they found, as we guess, there is no any kind of life there, but actually they found there has microbes everywhere. So if we can do this kind of research and get the enzyme, which will be very stable, even though in the very high temperature, it still can work. So this was a very good extreme environment to do the research. Of course, similarly, if we can do in the volcano, that will be also very new research we can try in Indonesia. Actually, from this environment, many compounds already be treated or isolated from different environment. In our institute, there is a group which only do the North Pole and South Pole. They most do the new species, but didn't do any chemical research. In my group, actually, most was focused on the Chinese extreme environment. Like in Xinjiang, we focused on Lobnauer, that's a soda lake, which already dried up for many years, and also the Taklamakan Desert. Then also in the west of China, there has a lot of cascade. So this environment was also very unique. We also put the energy to do research on here. Of course, there are not so many mangroves in China. Indonesia is the #1 mangrove species and areas. And also we go to the Tibet, Chenghang Tibet plateau, which is very high. We so-called the third pole of the Earth. We also collect sample in that area. So that's most we have done in Chinese environment. So of course, we recently get many very good species in those areas. Except in China, we also cooperation with many different countries. I've been 3 big islands. One was Kalimantan, which we has ever cooperation with the Sabah University to the mangrove in that area. And also we cooperation with Siberia scientists in Russia to the Soda Lake in Siberia. And we do the cooperation with the Pakistan and do the desert, Thar Desert in the border of India and Pakistan. Actually, when we do all those research, we get many good friends. And from different countries, actually, we get more knowledge about the research progress in extreme environment about on the microbiology. So now I give 2 examples which I have done in my laboratory. When I come here, I saw our institute research center. They has cooperation with the Cascade University. In the very beginning, to the Cascade microbiology. So I put this as an example. Another one was from desert we have done during past 10 years. So after those examples, our colleagues in Indonesia, we can know what we can do and hope we can have some cooperation no matter in microbiology, in chemistry or screening model. We can see the desert map, which most focused on the Africa and Middle East. Also in the South, in the Asia, Central Asia, also many desert. But you can find in Europe almost no desert, very, very few. So Europe's, but in the very beginning, Europe scientists, they do the desert research with cooperation with the Chile scientists. They found a place, name was Atacama, which was in the Peru, Bolivia, Argentina, and Chile. So they do the research most in the Chile part. As it is said, Atacama Desert is one of the driest place on Earth. So in the very beginning, very few people focused on the desert. They think desert was like a poor nutrition place, very dry, and sunshine serious and X-ray everywhere and long time. But actually recently, scientists in this Atacama area, they select the most dry up place and collect stone and found how microorganisms can inhibit there, can grow there. So for example, there is no water. Where water come from? So they found actually they used like CaSO4 combined 2 molecular water. So in the evening, these inorganic compounds, they absorb water. Then microorganisms can use this water. And in the morning, they just evaporate it under very high temperature. Because the high temperature and the low temperature in evening, so the water will be concentrated by those inorganic compounds, which provide water for the microorganisms. So I visit our genomics lab, and we found there is many equipment which can take this kind of picture. So how about volcano? If there is a sample, can we find volcano? There is a kind of strength in those stones, so which give us some inspiration. So even in this high dry place, still a lot of bacteria you can find. So how about do the research on secondary metabolites, which was the candidate for antibiotics? So you can see from here, if we put Atacama Desert and actinomycetes put into the PubMed, you can find only after 2008, then some research was beginning. Here now, only like totally 6-8 papers. So very few research on this field. Actually, this field was the most studied by a group scientists from Europe. For example, ERA has been the Tübingen University. So the Hans Fiedel, actually very famous on the, already retired, very famous on the secondary metabolites. And United States, Newcastle University and the University of Kent. These two scientists was very beginning to have this kind of idea. Let's go to the dry space to find the antibiotics. So they combine with the Chile scientists, with the chemistry, antibiotic, or also with a microbial chemistry. So they get together, begin to do the research in the Atacama Desert. Now they already found many compounds and also published a lot of papers. You can see most was from 2010 to 2023. New species and new compounds. Actually, Chinese scientists follow all those research, also invite them to do presentation in China. So Chinese has like a scientist field has this kind of meeting, Fragrant Hill Science Conference, which was a top discussion place for new idea or innovation technology. So we invite all those scientists which focus on the extreme environments and give the topic. And we, like Tony said, introduce Indonesia extreme environment. We introduce all the Chinese research and the environment and hope they can give some suggestion. So at that meeting, we learn a lot. Actually, they are pioneer for this field. What we do was actually Taklimakan Desert, which from the literature, they said this area was very similar with Mars. So even though we begin to do the actinobacteria in desert in 2019 or 2011 or 2010, actually this research already began. That's by the scientists in Mars area. They always think in Mars, are there any life there? So they try to find microbes in that area. And they think in the area of desert, they has the desert storm, and which was very similar with the Mars. So they prospect or they predict there's an environment of desert in the Mars. And they compare the picture, which was taken by the satellite camera, and compare with the most extreme desert in the Earth. They found many places was similarity. Of course, they go to those deserts to do many experiments, which was far more early than we do the antibiotic research. Kim, actually, he was a NASA scientist. He was the first people to set up the Atacama project teams. He visited so many places in the Earth, which was an extreme environment. He not only found Atacama, Namibia, and Death Valley. Also, she found Taklimakan Desert in China, also very similar with Mars. That's one reason we do this kind of research. And in 2011, we begin to do this research. in the Taklamakan Desert. Now we also begin to do the China Desert, this area, in the Tibet at Chenghang Plateau in the 2023. So now we have two group students now focus on different deserts. Actually, go to those places is quite difficult because there are no people and the local language we cannot understand. When we do the Taklamakan Desert, I'm very fortunate because I have a student from Chinese, but she was a Kazakhstan. She can say Uyghur and also can speak Kazakh. So that area is a lot of Uyghur, so we can very easily discussion and talk and do, for example, find loads. From Atacama Desert, we used like more than 11 selective media to get all kinds of actinobacteria. So many new species and active strands was found there. This I just give some example. We are already published all those new species found in the Taklamakan Desert. Also many active strands. From one strain, which was the Micromonospora, we found new antibiotic, which was very strong activity against gram-positive, which was drug-resistant strains. So this work actually we take us too long time, like 1 year we elucidation the chemical structure because there are a lot of carbon which cannot be very well like settled down. And finally, we elucidation the structure, which was the CO peptide antibiotic. This antibiotic was divided according to this part of structure. If they have several different kind of chemical structure with nitrogen, so we belongs to breeding this series. And after we finally settle down elucidation the chemical structure, so we found we have our compound has many difference with non-compounds. And we have 4 sugar moiets. Most was none or only 1 or 2. So this group antibiotic, which has a short page because they are too difficult to dissolve in the water. So we more sugar moieties will help they are water soluble. And very fortunately, the yield is kind of high, but also we found some small one. So we put all our energy, finish all those structures. But finally, it proved the Shenzaomycin A, the biggest one, actually was more active than others. You can see in the vitro environment, the activity was like 0.6, 0.25, 0.23, which was quite low against all those vancomycin drug-resistant bacteria. Of course, this compound was more active than vancomycin. We found if they lost this moiety, like it become Shenzaomycin B, which lost this moiety. So the activity will be less strong than Shenzaomycin A. So we use the Shenzaomycin A, go to the animal test. Very good observation. Actually, there are no mice died, and ED50 was 2.97 microgram per gram. So vancomycin was 4.48. So we are much better than this one. The only thing which this compound need to improve actually was water soluble and also yield. Now our institute, our laboratory already begin to to say how it can like genetically biosynthetic in E. coli. Also, we cooperation with different group. We try to improve the yield. We have used many methods, and some has improved a little bit, but some method was good. But now, still, we didn't get a strain can go into the commercial, can go into the factory. So that's what we still need to do. After this research, we accumulate a lot of experience on how to do the research in the desert. And we cooperation with the Mongolia, which outside of China, that place was Gobi. Gobi is also a very big desert. We cooperation with the Mongolia scientists. And also we have cooperation with Pakistan, Lahore, that's the Punjab University. And we go to the Thar Desert, do the research. The students come to my group. We get together to do this kind of research. Now the Kizokum and Karakum, which was in the Turkmenistan, Kazakhstan, and Uzbekistan. Also, now we cooperation with Uzbekistan scientists. We now do the research in this area. Yeah, Pakistan, actually, they also go to China to do our Kup Desert. So we exchange sample and do many good research and publish many papers. And their students go to our lab to learn how to isolate new species and do new species identification. And now more than 30 papers we published, marked with red, with cooperation with different group. And there is almost 5 countries work together. From astrobiology, they recently have paper about hyper-arid Chidam bison on Tibet plateau, which very attractive. We have done this area. This area we have never done before. So from this paper, they give some a picture which was really dry up and quite similar with mouse. So in the 2022, I was invited by the Chenghang University, and they give me some grants to do with their scientists. So we, in the 2023, we begin to the Chidam Desert. This area already collect samples, so I will show a little bit about the place, which might be you never seen before. That's the Yatdan, which was the mountain in the desert. Finally, was decayed, and we collect sample from the Yatdan, very dry place. When we collect sample here, we found, okay, almost very, very few water. You can see all was like inorganic compounds, which microbes are very difficult to get nutrition. Now we already begin to do this work. Now we talk many about the desert and research. Another work actually was almost 5 or 10 years before. That's cave microbes and antibiotic discovery. So I have get the certificate to do the caving exploration in 2011. And I go to many caves in China, and that's one picture I have taken in the cave. So you can see the snail, which was so small, and they has transparent. They changed totally. They have no any color in their body. You can see the yellow one. Actually, they eat those mud into their stomach. So you can see that's the yellow one. But from if give something clean, you can see it's transparent, no color. So to the cave actually was more dangerous than desert. you need to learn many things and get familiar with all that equipment, just like diving, which was really a dangerous work. If diving in the cave water, that's more dangerous than diving in the ocean. And usually, if you do the cave, because I saw our institute, our research center has cooperation with the university who was a cluster cave microbiology. So, I give some my suggestion. Actually, if you do cave, usually cave connected with water. There's no water, no cave. So you usually will get sample which was old sample, was out from a cave, not cave local sample. So if you want to get those samples, you try to find a place which was dry up totally. No water go to there because water will carry some samples outside of the cave. So that's why we have to training ourselves to go to the deep, go to the survey by ourselves. So many of my friends say, Chenghang, you don't need to go to the cave. I can collect sample for you. But I know when I get the sample, most of our energy was wasted because they just get some they don't know where we can to find a good sample. Actually, cavings spend a lot of money because the lobe is very expensive because they are closely related with the life. So the lobe is not good, and they touch the rocks, and it's easy to like broken. So we should take more care of. And also we need a group of people, not only one or two people, to do this exploration and survey. This was Guizhou, actually Chongqing, which was in the west of China. They have name of kingdom of caste. So there's a lot of cave. And these two students of my PhD, actually, we already explore and get a very good way for them to go inside. So they do the young sample collection and do the micro research and directly in the cave. We don't need to hood because in the cave you find a place which was no water coming, it's no wind, so very clean place. In the cave, actually, we get a lot of strands. There is a strand which you can see here, actually directly inhibit all others. So we select this strand, and we found it's a streptomyces. And because it's a very strong activity, so we first didn't do many polyphasic taxonomy. We just direct go to the chemistry. So we found this antibiotic, the compound can kill many like not only bacteria, also cancer cell. After we purify the compound, actually it's not easy because when we do the fermentation, we need to totally dark. We need to, it's not stable. We need to avoid any light. And finally purify these compounds. And we get 3 compounds, which even though it's already have a similar structure of compound, but the difference is obviously most this kind of group of compound has amino sugar. They have nitrogen, but this was only one, no nitrogen in this sugar molecule, which was very unique. That's why some like Nature product reports, they analyze our structure and point out the cave is a good place to do the research. After we purify the compound, and my colleague, also a Professor he do all the anti-cancer research in vitro and also do the mechanism how the compound kill the cancer and get clear about how to kill cancer by what kind of mechanisms, which was not in my research field, but with cooperation and get good results. Shacamycin, this compound actually was number 3, the 3rd new compound in the world. It's the very beginning, the first compound in the Asia from the cave. So this was fourth. So I follow this research till now. So not many, because it's too difficult to get sample in the cave. Also, what I should say is get good sample in the cave. Okay, I've give the 2 examples which we studied in the extreme environment. And it's very fortunate I met Indonesia scientists. I met particular university students, rector and mangrove expert in 2018, and 2019 I visited their university, and their teacher, like lecturers, go to my lab to do the postdoc for 1 year, and we get grants, and then I can visit Indonesia and met Dewi, met many of my colleagues in Indonesia. It's lucky for me to have the chance to do more research together. So here I introduce some international collaboration grants, which between Indonesia and China. So now many chance, much more than before. Actually, I introduced these 2 type of grants. Hope I can success to help Indonesia experts cooperation with Chinese scientists to do statistic research. There is 2 types. One was bilateral cooperation. That means 2 countries, Indonesia and China. Another was a multiple lateral cooperation. So at least 3 countries should be involved. The first one, bilateral collaboration, actually most in Chinese part was founded by Nature Science Foundation of Beijing and the Nature Science Foundation of China. There are 2. And also the Ministry of Science and Technology of Chinese people, China. In the NSFC, regularly, every year, they will have an announcement for foreign scientists to research in China. Only you need is a host, like I come here as a visit, like scientist. Ira and Dewi host me here, so there has institute can host you, and your leader like Tony can prove that. So in China, the same thing. For example, you want to apply these grants, go to our lab, we can host, and our leader agree, then you can apply it. So there is several different. One was for young scientists, some for like excellent and also senior. So they have different type. So the grant also different. Then usually 2 or 3 year grant, including research budget and the living budget. That's an SFC. And also recently Beijing, they are not regularly. They were sometimes suddenly they have some grant appears. For example, before I come in the August 8th, suddenly I get the information and they announced they have built a joint laboratory with Indonesia. So even though 14th I need to take flight, so only these few days I write the grants and frequently discuss with the Indonesian like Devi and Ira how we write what kind of things. So that sometimes they give the announcements. And that's even though I was here, they are very quickly let us all the defense. Now we finish all the defense and waiting the results. And another one actually was multiple. for example, more than 3 country scientists need to involve, like Asia submit participate countries, at least 3 countries, we can apply e-Asia. This project always has, but sometimes they will change the research field. And also 2025, in the January in Brasilia, I remember in Brasilia, Indonesia has become member of BRICS. So now the BRICS has expanded. Before only 5 countries. So I have already applied with Brazilian scientists, India scientists, and Russia scientists ever twice apply BRICS. But it's kind of difficult, the competition serious. So actually now, since Indonesia already joined that international organization, actually we can pay attention to that, because this grant very high impact in internationally, very high impact, and the funding period and money is much more than other. So I suggest our research center can pay attention to it, because it's just beginning from 2025, Indonesia as a member of this international organization. Another very important one was MOST, which always cooperation with BRIN. So in the BRIN, sometimes we can check this grant is very good, because they was like this: Chinese government give money to the Chinese scientists, Indonesia's government give the research funding to Indonesia scientists. So we not only can use that money to visit and exchange idea and cooperation, we can do more research than before, and we can do some research which was very difficult. For example, like this year, 2025, this MOST grant already finished. They say Intergovernmental Cooperation of Science and Technology. This grant actually have to sign agreement between 2 countries, like China and the Indonesia government. They already signed a contract or sign agreements to do this kind of research. But every year they are changed, the research field. For example, 2025 actually applied microbiology, which I check your institute research field. Actually, this was suitable, like food and agriculture, like food biotechnology and milk production, which is a kind of good for our institute. Usually, they only 6 projects will be found. Totally, they have 3 research fields. So that means each one has 2 grants will be supported. Usually, they has 2 years, but funding is big funding. So I really think it's a very good funding. Our Indonesia colleagues should pay attention to that. Actually, if you have any information you need to, so you can give this email or telephone number, you can ask them and pay attention to that. Also, when I started here, and also Chinese government also give another funding, also a very big one. And this funding was matched with International Cooperation of Science and Technology. The money was for communication. That means scientists in Indonesia, they can visit research group 3 months, 6 months, and 12 months in China. And they focus on biodiversity of micro or other biology. And also like biology, medicine, space shuttle, low mate control, all those research fields. They have 10 projects can be supported. A very good one was they don't need many other requirement. For example, you must have PhD. No, they just say you should have PhD or research experience 5 years before, after 5 years. So, and also age is good, 45 years old before. So each month they will give you support for your living. This was from most. And also now still running this project, which was cooperation of science and technology. They have three research fields. One was for poverty alleviation by science and technology. This one actually was cooperation with Chinese scientists. For example, you have products want to try, for example, crop breeding and also green efficient culture. Then you can, for example, I want to try. It's helpful to the farmers, so they will support and give money to do the research. So this now still running. I'm not sure what's deadline, but now still running. This was recent grants in China, and also I have kind of information because some colleagues, they have students if they want to do the overseas study, and in this I have here was covered. We have a website, so we can register and we can search which university, which research field. That's for the young students. And I have checked our website, and we found there is a lot of research which was very matched with our research direction. Hopefully, we can have the opportunity to get together to do more works. And also, hopefully, if you want to find some cooperation in China, I can become a bridge to help to set up the relationship. We just talked about the most like grants. This year, actually, we apply grant with the Uzbek scientists in most. So because competition kind of serious, so our institute cooperation with 2 universities in Uzbekistan and do 2 directions. One was discovery of leading molecules and multi-target mechanisms against metabolite diseases like diabetes based on native medicine resources to China and Uzbekistan. So these are medical resources, not only plants, because Uzbekistan people, they do the plants, we do the microbiology. So we can cooperation together to do the research on that field. Also, we apply another one was identification of antimicrobial compounds with novel mechanism of GAT based on medicinal resources of Uzbekistan and China. That's our cooperation study to combat with drug-resistant pathogens. So this was another research group, and the ERA has no Dora. Actually, she was the overseas student in China. She helped us a lot as a translation and translator. And when we write all those proposals and all the language problem, Dora helped us a lot. Yeah, this almost finished my topic. Most work was done by all PhD master students or the overseas students. Each year, our laboratory hosts like 2 or 3 overseas postdoc. And also, thank you so much for Research Center of Applied Microbiology. I'm very happy to work here and have get very good friends. And I observe we have very, very good students here. And hope we can set up more long cooperation and do fantastic scientist research. Thank you very much. This all for my topics.
Okay, thank you very much, Professor Sun, for the insightful presentation. Now we will begin the question and answer session. To start, I would like to invite our participant. Please free to raise your hand or write your question in the chat box. Okay, there's a question maybe. Okay, while waiting, maybe I ask some first question. Professor Sun, you mentioned microbe from extreme environments as a promising source for antibiotic. Could you please elaborate on which environment you find the most promising for future discoveries?
Ila, would you please repeat it again?
Okay. Among the extreme environmental you have
Oh,
studied, yes.
uh-huh.
Which one do you find most promising for discovering novel antibiotic producing microbes and why? Most promising, yeah.
Yeah, actually, usually when we get the streptomyces, we think too many people have already done the streptomyces. So even though the activity is very strong, we still put them in the second. So the first one, we want to do the real actinobacterials, which has strong activities. So we have which one was priority to do the research. We have something in my mind, but sometimes it was really lucky or not lucky, even though the strain was very new species, and has activity. But when you do whole genome sequence, then you will find it's only kind of very few antibiotics there. Is far less than streptomycins. So streptomycins still very good candidates if they come from unique kind of environments. And also one thing we should consider is the yield. Sometimes the activity was very strong. As you know, usually the chemical structure from the microbes is very complicated. So chemical synthesis is very difficult. So how to get yield or enough amount to do animal tests, which was what we should think, because otherwise we only publish paper. So we cannot do any further in animal. The in vivo activity is very difficult to carry out if the yield is so low. So when we select strains, we think a lot of things about, because once we decide this strain, we should do the chemical study, then a lot of energy we are put inside. That's a priority we need to think about it. I'm not sure, is that okay for your question?
Yes, thank you, Professor. Okay, let's move to the next question from the chat. I think I saw some question. Wait. Okay. From Nurfianto Giar Paninggung Malang. Question to Professor Chenghang. What are your view on synthetic biology approach for applied microbiology? Also related study for microbe from the extreme environment and its utilization, such as synthetic microbial community. Are these syncom already widely used in among researcher and industry? Thank you.
Yeah. Actually, now synthetic biology is very popular, and in my lab also we do this research. Which was like what's your target for your research? Actually, in my institute, get the medicinal drug was number one. Published paper was number two. So if you want to publish a very good paper, right now the synthetic kind of biology is a very good direction. And if we use the synthetic approach to dissolve the problem, for example, the Saccharomycin, they have a lot of sugar. In the sugar, there hydroxygen. This hydroxygen was covered by the methyl. So the water solution is still very difficult to dissolve in the water. So we want to cut methanol from hydroxygen. So we used like synthetic approach to do that work. Is very sensitive, or was meaningful, and can help very much. So now, like genetic mining and biosynthetic approach, which was very good, and how to say, modern technology. So we in my topic, I didn't touch much of that. Because we most was in the extreme environments. But this one was for modified compound and improve the yield, which was very useful. So biosynthetic approach can divide into two parts. One part you can think about chemical synthetic. The function was almost the same, but the way was different. And now was basic research, used the synthetic approach. So that's the answer. I'm not sure is good or not. Is it satisfied?
Okay. What about this? Mr. Nurfianto, this is satisfied for you, the answer from the Professor Chenghang? Or you have another question? Okay. Think Mr. Nurfianto not respond. Okay, let's start with another question. This is we have here from Evita Evita Krishna Yanti. Professor Chenghang, do you use specific media to get or isolate unique actinomycetes, or do you just use common media like ISP2?
Oh, we usually we have used more than 10. So actually, we can find ISP2 is not enough. ISP2 usually we use it as purified. For example, we already get a strain, we want to purify it because it's a very universal media. So we, in our paper, usually we used more than 10, and we found the more media we select, sometimes we use not only like ISP2, then we ISP2 plus different pH and different salt. So they will also change some selection, change some diversity. So that's why sometimes our students work really hard on those kind of work. They have many plates. So if I suggest, please use more than like at least more than 8. To you can find from the many papers they have different recipes.
Okay, thank you, Professor. Now I would like to invite Budi Mediana to ask the next question directly. So Budi Mediana, just ask your question, please. Please unmute your audio.
Okay, I'm ready.
Okay.
Good afternoon, Professor Sun.
Hi.
Hello, my name is Medina. I'm curious about your collaboration instead. During your collaboration with various institutions, did the collaborative team only focus on finding extremophile for the pharmaceutical purposes, or did they go further, like try to find microbes or for enzyme industry other than pharmaceutical, or maybe other microbes for environmental cleanup purposes potential during your collaboration with many institutions? Thank you.
Yeah. Yes. So actually, we cooperation with like more than 10 countries, and different research group. They have different purpose. For example, some was want to train the students. Some wants to like learn some technology, and like from Punjab University in Pakistan, actually we cooperation very how to say deep, because we not only do exchange students, also we collect sample each other. Then we do the actinobacteria new species and compounds. What they do was do the dyeing, DYE dyeing, different color. So so when we get like dyeing strands, then we transport to them. When they found some active strands, they transfer for us. So usually students do this kind of research. And also like enzyme, you know, the biosynthetic way, most should be confirmed by the enzyme. So in my group, also researcher, they knock the gene or they cut the gene and they express those enzymes and use the enzyme in vitro to synthesis. So to prove the enzyme actually was the truly participate in the reaction. So so the different research field, different like work. Also, we have ECTA to purify the enzyme, but we didn't like, for example, in the applied microbiology, they like thinking about how to get enzyme from some strands which can degradation or do other work. We didn't do this, but we can. We know all those research fields. Only different was the assay model. For example, you want to, like in the mengundang group, they get the strands which can degrade the PVC and try to find which enzyme was take reaction. So we can do that. Only the assay model was different, but we not put more energy on the enzyme we put in the small molecules.
Thank you,
Yeah,
Professor.
like this, yeah.
Okay. Thank you, Professor Chenghang. We have another question from the chat from Faika Umar. Professor, many extreme environment microbes have been studied for decades. What criteria distinguish new discoveries from incremental rediscovery of already characterized traits?
Yeah, actually, it's depend on money. Actually, if we can do the whole genome sequence, and then we can blaster, we can find the like slightly difference in some whole genomes. So we can, if they located on the whole biosynthetic genes, actually we can see this compound might be new, not totally new, but some place will be modified because they have different genes. So that's also the way we are thinking why we go to the cave. Because, for example, the cave, they have no light. So some gene will be changed. They don't need to sensitive the light. So they delete some genes, and so the whole genomics changed much more than others. So they can biosynthetic new compounds. So that's we insist extreme environment, especially the life condition is completely different, like a super dry desert and cave. So I think might be in the like deep marine if we get sample during the like black chimney, that will be very good to do the research.
Okay. Thank you, Professor Chenghang. We have another question from the chat from Malik. How is the relationship between bioactive compound which produced by bacteria and the chemical compound on environments, and are the kinds of antibiotic effect by microbiomes?
You mean the co-culture of microbiology?
Which produced and the chemical compound of MK. Maybe the relationship. How is the relationship between the bioactive compounds and the chemical compound in environmental?
Environmental. Active compound. I repeat the question. So that's when we do the fermentation. So the fermentation condition will change the production of compounds. Is that what I mean?
Yeah, it could be maybe just the effect of the antibiotic in the environmental, maybe.
Okay.
So the relationship between the bioactivity and also the microbiome, maybe this question. Or maybe Mr. Malik, do you mind to talk with Professor Chenghang in more detail?
Yes, please.
Mr. Malik, do you mind to talk with Professor Chenghang directly? Oh, the signal is not stable in Mr. Malik. So maybe you can also read in the chat. How is a relationship within bioactive compound with produced by bacteria and the chemical compound on environment, and are the kind of antibiotic affected by the microbiome?
Like this, for example, if we want to, usually we will set up a screening model to screen bioactive strains. Then, if that strains produce compounds which was bioactive, then we will identify which kind of species. If it's a real or new actinobacteria, so we will think more about to do these strains. Actually, by the way, we sometimes can find very unique phenomena. For example, the bacteria, they grow 11 days. When they touch the fresh strands, they will kill the fresh strands. So that says a self-defense kind of strands. So during your research, you will find many different kind of phenomena. So if this kind of bioactive strands we found, then we will continue. We will not take care of about its yield or its stable or not stable. We will, because it's a very interesting strand, so we will directly do the basic research of that.
Okay. Thank you, Professor. Mr. Malik, do you satisfied with the answer?
Hello.
Yeah.
Can you hear me, please?
Yeah, I can hear you.
Yes. I'm satisfied for the answer.
Okay, thank you, Mr. Malik.
Okay, thank you.
Yeah.
You're welcome.
Okay. We have another question from the chat from Anggia Prasetya Putri. Professor, sometimes isolated microbes stored for a long time can lose their ability to produce specific active compound as the storage conditions may not provide similar external pressure as the extreme environments from which they were isolated. Have you ever experienced this, and how do you ensure that this does not happen?
Yeah, actually, this really happened. And
Yeah.
for
Sorry.
example,
Yeah.
you have a very good strain, and they produce something. Then a long time later, you want to use it again, you will find okay, they just disappear. Then you go to the sampling place, you want to isolate the strain once more, but you cannot anymore. So that really happened. So how to say, if you take the sequence, the biosynthetic gene was still there. So that means it's not, you didn't, for example, after you contaminate it, you have to purify it. Sometimes you just purify the contaminated one. The original one you will don't use. So that will be a problem. Another problem is when this strain was contaminated with other strains, which they are always close together. Then you want to like fermented the compound, you cannot anymore because the strain already not pure. So there is a paper from my colleague. Actually, they analyze streptomycins, which has like spores. So they used one strain which was fluorescent genes. They co-culture. Then they will find the streptomycins in the spores. They have some luminescence. You can see the light. That means the strands already go inside the streptomycins, which you use these strands to do the fermentation, you cannot get your compounds anymore. So they have several different kind of reasons. The most reason might be it already been contaminated. If you are resuscitation is success, but it still cannot produce, might be the already been contaminated. So you can select like more than 10 different selected media you can store there, and you can finally separate these two strands. Then you will success. I met this kind of problem.
Oh, I see. Okay. Miss Anggia, do you satisfied with the answer?
Hi. Hello. I think I just wanted to clarify a little. Can you hear me?
Yes.
Yes. Thank you for the answer. I was just wondering if you know for sure that the microbe is pure and you've ruled out contamination, but because it's not there's no external pressures anymore, so the bacteria just does not want to produce that compound anymore. So is there anything that you can do to revive the ability to produce that specific compound again, or that's it? There's nothing else you can do.
Yeah, you can try. Yesterday I talked with about my professor take sample and they take strain go to Japan. They cannot ferment it anymore because of water. So Japan's water with Chinese water and with another like industrial factory, the water was different, the yield was different. So microbiology is very sensitive sometimes. So before you are medium, or you just for example, you buy some medium, this medium and that batch of medium, then the composition, for example, was different. For example, your fatty acid, they are different. So sometimes will affect your production. So the way you have to do was you select more media. Like, you can try more. You will success might be. We met these kind of problems.
Okay, so you're trying the different media to see if you can trigger production of the original compound.
Yeah, that's right. Yes.
Okay, thank you so much.
Thank you, Professor Chenghang. We have another question from Eri Sahabuddin. Professor, can extreme morphils provide solution to multi-drug resistant challenge? Have you tried the active compound or antibacterial for multi-drug resistant?
Yeah. So actually, I didn't talk much about drug-resistant bacteria. In my lab, we not only cooperation with hospital, which was drug-resistant bacteria, we also cooperation with hospital, which was fungi, drug-resistant fungi, and the new appears fungi. For example, now some like HIV patients and cancer or to the operation patients, their immunity is very low. In the hospital, it's really easy to infected by the drug resistance bacteria and fungi. So actually, a large part of research, we focus on that research area. And also, like Ira, you have done the Pseudomonas aeruginosa, which if it's a drug resistance strains, it's much more difficult to eliminate that bacteria. So yeah, many energy was focused on that.
Okay.
Is that
Okay. Oh, yeah. Yeah. Mr. Ari satisfied with your answer, Professor. Okay. Next question from Ira Puspita. Professor, I isolate extremophile yellowish bacteria from geothermal, but the bacteria has no spora based on literature. Then in a short time, the color changed from yellow to white, and I'm not sure it is the isolate or another. So it's mean we lose the isolates?
Yeah, the bacteria with the glows, if it's pure, they change color is normal. It's not change color, it's they produce some pigment into the into the media. So the strain in the very beginning is a kind of a morphology. Then after 1 week, they will be changing another morphology. It's normal. And also, for example, in the very beginning, they don't produce pigment. But 1 week later or 10 days later, the whole agar was black. It's possible. It's not your strain was unpurified or some reason. Actually, they glows different stage. They have a different morphology.
Okay, I see. Do you have another question, Miss Ira Puspita? Miss Ira said that, but I never get the yellow again until now for many times subculture. What happened with this one? Yeah.
Actually, the pigment is very interesting. Never get yellow anymore. Okay. That's what reason. So you subculture in the same media or subculture in another media?
Yes, Prof. Sam.
Okay.
Yeah.
That's bizarre. Usually, then it's a subculture, then purified, or one strain was like endophytic together, then another was died, or after several subculture. So that one was so weak. Yeah, that's actually if there is this kind of phenomenon, it's quite similar with co-culture, this kind of research.
Okay.
Where the strain come from? For example, they come from like deep sea or some environment. So when you culture them in the black place or have some UV, they will be different.
Okay, Miss Ira, do you mind just ask directly to Professor Chenghang?
Oh, okay, thank you, Prof. Maybe I will try to another media because I just use NA, just general media, and I just realized that this is need a tryptophan on any and yeast and
Okay.
stuff like that.
Yeah, actually, do the microbiology is happy because you always met different kind of phenomena which was attractive your attention.
Yeah. Thank you.
Thank you, Prof.
Yeah. Is there any question again from the floor? Maybe somebody want to talk directly to Professor Chenghang? Okay. Okay. If there are none, I will go ahead and close today's seminar. Okay. Thank you again, Professor Chenghang, for the enlightened session, and thank you to all participants for your active engagement. We hope you find it insightful and inspiring for a future research collaboration. Finally, I would like to hand the program back to our MC, Bu Dewi, to continue with the next agenda. Bu Dewi, the time is yours now.
Ya, makasih Mbak Ira. Thank you Mbak Ira for leading the seminar, and I'm so happy that there are a lot of questions, and it's mean that many of you are interested to the topic that Professor Sun just presented. So if Professor Sun still will be in our laboratory, Taxa laboratory, till Friday afternoon. So I invite you, welcome to the Taxa laboratory and meet Professor Sun face to face to discuss the topics that you are interested to, or maybe you want to asking about the collaboration or funding that you can apply to China government. Are you available, Professor Sun?
Yeah.
For the next 2
Warm
days?
welcome. Yeah, warm
Okay.
welcome. And also, I will hand over the PPT to you. Or
Yes.
some people they need just get it.
Yes, I already make like a Google Drive, so I will share the link. So for the audience who interested to get the PPT, you can download for my Google Drive. So one more time, I want to encourage, especially for the young researcher, because in Professor Sun institution, there are many of scam to invite you to come to his laboratory. So the collaboration is open to all of you. Also, I the PI of the collaboration between our institution, but the collaboration are not only for me or for Ira, but for all of you. If you are interested, just discuss it with Professor Sun. I think this is the end of our webinar for today. Thank you again for your participation, especially for Professor Sun and also Bu Ira, who already lead the seminar. Sorry if there is a mistake for me of the organizing committee. Last, Assalamualaikum warahmatullahi wabarakatuh. See you again in next webinar. Bye-bye.
Thank you.
Bu Dewi Zilda, we have a certificate. Don't forget. Yeah.
So we can get it. Have
Yeah.
you seen the link to download the certificate?
Yeah. Actually, Bu Lia, do you mind to show the certificate for Professor Chenghang? This is from our research institute.
I mean, what about for the participant? Do you provide the certificate too for participant?
I don't
Maybe
know.
the link where we can where they can download the certificate. I think some of us need the certificate for this seminar.
Bu Lia, there's a certificate for participant also.
Yeah. Maybe we can ask Fathoni for his certificate. You can mail me on chat box. You can issue for the certificate. Like to ask for
certificate for this webinar, you can just directly email me. My mail is on the chat.
Would you share your email address?
Yeah, in the chat box.
Okay. So please, all of the participants, you can copy the email address and then contact Mbak Nur to get your certificate. So can we end this seminar?
Yeah.
Okay.
Yeah.
Thank you for all of you.
Yeah.
Bye.
Thank you so much, Professor
Assalamualaikum
Chenghang
Thank you very
and
much. Thank you for hosting.
everybody. Bye.
Bye-bye.
Bye.